Massimiliano, for your presentation, and thank the organizers for allowing me to do this presentation. MNGIE is a disease with these clinical features. I mean, the main debilitating feature is gastrointestinal dysmotility, which leads to cachexia to the patients. It's an adult disease, although it's a severe disease. As you can see here in the survival curves that were published some time ago by Caterina Garone and by the group of Van Lombes. I mean, in most cases, patients die between 20 years and 40, 50 years of age, so it's a severe disease. There is also peripheral neuropathy, also affectation of the extraocular muscles, and as I said, it's a severe disease, although there are other mitochondrial diseases that present earlier. MNGIE is caused by mutations in the gene encoding thymidine phosphorylase. Thymidine phosphorylase is a nuclear gene and is a cytosolic enzyme that degrades, is fully responsible for degradation of thymidine and deoxyuridine. And if thymidine phosphorylase doesn't work, what happens is a systemic elevation of thymidine and deoxyuridine in the whole body, in the whole cells and in the whole water volume of the body because they are water-soluble molecules, thymidine and deoxyuridine. In normal conditions, those tissues and those cells that express thymidine phosphorylase get rid of the thymidine generated by all tissues that express thymidine phosphorylase and tissues that do not express thymidine phosphorylase, because the thymidine phosphorylase go to the circulation and then is degraded by liver and by hematopoietic tissue that expresses thymidine phosphorylase. In MNGIE patients, since you don't have thymidine phosphorylase, there is a systemic elevation, and it is important to understand that is the systemic elevation the cause of MNGIE because it also affects to tissues that usually don't express thymidine phosphorylase. So even if you normally don't have thymidine phosphorylase, you are affected by this elevation of thymidine. And the mechanistic details are the fact that elevation of thymidine within mitochondria is phosphorylated by TK2, which increases the levels of DTTP, but also secondary decreases the levels of DCTP, and this imbalance of the mitochondrial nucleotides produces mitochondrial DNA depletion, multiple deletions, and in this case also somatic point mutations that are produced in the patients but are not inherited from the mother. And this, at the end, results in mitochondrial dysfunction. And since the cause of this disease or the mechanistic triggering of this disease is the elevation of thymidine and deoxyuridine, the therapy approaches since the beginning were aimed to reduce this systemic elevation. And the first thing we tried to do was hemodialysis. But the hemodialysis, as you can see here, transiently reduced a little the concentration of thymidine, but very fastly, because there is a fast turnover of this molecule, thymidine increases again. It was also tested in some patients, in some reports, peritoneal dialysis. And in this case, it is curious because the reduction of the thymidine at the systemic level was not almost significant, as you can see here in this table. The reductions were very mild. But patients noticed some, in these two reports, noticed some kind of consistent improvement, especially in the gastrointestinal symptoms. And this, I will talk about this later, may be related probably with some kind of local effects. I will talk about this later. I mean, this is a theory. It's nothing that we know, but we have to consider that. Then there were other approaches in which there was enzyme replacement, not removal by chemical means like in the previous strategies, but putting in the patient thymidine phosphorylase, doing enzyme replacement. One of them was developed by Bridget Bax in the University of St. George University, I think in London, by putting thymidine phosphorylase in the erythrocytes of the patient and reinfusing. So they took the erythrocytes of the patients, they put thymidine phosphorylase inside, and then they put it again in the patient in order to degrade the thymidine and the deoxyuridine. And you can see here that there is degradation, but this degradation is transient and also not permanent. So you are not achieving what you want, or you are not fully achieving what you want. And when in a further paper in which I think they did it with more often, there is probably more reduction, but still is not permanent because these levels here are levels that are practically MNGIE levels. We also did platelet infusions transplantations to patients because platelets has a lot of thymidine phosphorylase, and we found similar results. I mean, there is a transient reduction, which is not sufficient, but in this case, this served as a concept that probably bone marrow transplantation could be a good option, because in this case, it's not that you are infusing platelets periodically, but you are providing the whole hematopoietic tissue to the patient from a donor, and then the thymidine phosphorylase there would work. So this was the first sign or the first results that prompted to do bone marrow transplantation. So I go now to the therapies that now are in use for MNGIE patients, which is the first one, allogeneic hematopoietic transplantation. As I said, donor-derived white blood cells will provide, and platelets will provide the TAP enzyme and reduce the thymidine phosphorylase, but also liver transplantation, in which liver, which also expresses a lot of thymidine phosphorylase, will reduce the nucleosides. Regarding the bone marrow transplantation, there was a report published in 2015 run by all the collaborators and all the teams that had done transplantation, and as you can see here, effectively, I mean, the thymidine phosphorylase in the blood of these patients went up because you have the graft from the donor that contains thymidine phosphorylase. The reduction of thymidine was also effective, also, although we have to consider here the levels that we are measuring here. I will go to this later. And then clinically, there was a positive effect, but very slow. You can see here, probably, if you don't see it, this scale here is years, and this is 4 years. So in 4 years, the body mass index increases, but very, very slowly. So, and the same for nerve conduction studies and other studies here. So there is improvement in the patients, although it's very slow. So sometimes the teams talk more about stabilization of the disease rather than a spectacular improvement on the disease. Stabilization is good because this disease, without treatment, especially when the symptoms start, is very degrading, and at the end the patients die. So stabilization is a very good thing, but it's not something that reverses rapidly what has been produced in the patients already. But the main problem with this was the survival to the treatment, because the patients that survived, that were the ones that were shown before, don't hide the fact that in many cases the patients died. This is in this plot you see the survival curve, in which you see that more than 60% of the patients died in the first few years after the treatment, in some of them in the first months of the treatment, in many cases because of the treatment, because it's aggressive. You have to immunosuppress the patients, and then this is very aggressive for them, especially because they are in a very poor condition. And then, for example, in the second plot, we can see that there is a big difference between groups in which the match of the donor was full or when it was not fully matched. And also, depending on the fact that they had severe or history of pseudostructure, gastrointestinal pseudostructure or not. And then also depending on the intensity of the conditioning. So this was a therapy that was very, very aggressive, and then with a very, we could say probably unacceptable fail rates. And then, in 2014, one Italian group from Bologna proposed the fact that this disease could be treated also by liver transplantation, because liver contains a lot of thymidine phosphorylase. So in MNGIE patients, the liver doesn't do anything, but if you put a liver from a donor, you will remove the systemic accumulation of thymidine and deoxyribonucleic from the patients. And many patients have been transplanted so far. And then the results, I mean, this is a slide with a lot of text, and probably I cannot go into the details. But what is true is that even the patients that have been reported from Italy, and some other patients from the USA, and also the cohort of 7 patients that we have transplanted in Barcelona, in all the cases, thymidine and deoxyribonucleic are reduced, so the reduction is the liver works in all of the cases, and the clinical results are similar to the ones that we observed for the hematopoietic cells transplantation. There is a stabilization or a slight improvement. Many patients survived. In other cases, they died. For example, in our cohort, there was one patient that died because of the aggressive of the treatment, because the treatment is probably not as aggressive as the bone marrow transplantation, but it's still not a joke to a liver transplantation. So the results are similar, and well, I'm going to present a little bit something not so clinical, but a little bit more biochemical about the patients that were transplanted in Barcelona. And then here I want to go with a little bit more of detail what we achieve with this liver transplantation and also with the bone marrow transplantation. The level of the normal levels of timidina and deoxyuridine are below 0.05 micromolar. In fact, there are reports that have been obtained soon, short time ago, in which the levels have been actually measured and is 0.003 micromolar, meaning 3 nanomolar. And the levels that so far we have been able to measure are 10 times more or less. So in patients, the levels that you reach are 0.5 micromolar. So, and this line here, a small line here, is the limit of detection we have. So we are reducing, and this is a good thing because we are removing most of the cause of the disease, but we are not fully achieving normalization of time levels of timidina and deoxyuridine. And these are 4 patients that we transplanted, and then 3 more. So in all the cases, it's the same. The levels in these patients remain more than probably 100 times the normal levels in healthy people. Although in MNGIE patients, the levels are much, much higher. So we have to consider that, and we have to consider that probably trying to reduce even more wouldn't be a bad thing. In the case of liver transplantation, you don't reach timidine phosphorylase in the blood because you are putting the liver, but in the blood you have the same. It's true. I mean, these are not normal levels of timidine phosphorylase. This is timidine phosphorylase, as this is the time after the transplantation. This is 5% of timidine phosphorylase of a normal people. So you don't do anything, but at the beginning of the transplantation, just after the graft, you see a very transient small peak of timidine phosphorylase. And this can influence the fact that probably in the first part after the transplantation, you see something in vitro degradation. You take the blood, you leave the blood here, and you are reducing. So you are seeing some, I mean, the levels in the patients are not these ones, but are probably more these ones, okay? But anyway, this is probably not very important. I just wanted to present in our cohort of patients, there was one patient that was treated early in the course of his disease. And I think this is important because if you treat the patients before they start developing severe manifestations, especially gastrointestinal manifestations, probably the chances of success or being more effective are better. I'm not clinician, so I am showing something that probably I cannot tell you anymore, but well, these are nerve conduction studies of this patient, follow-up before the transplantation. This is the transplantation and after the transplantation. And you can see here, in many cases, not all of them, but in many cases, after a decline, after the transplantation, you recover. You have a tendency to recover. So this is just the patient in which we have a better follow-up and also that was less affected or was a little bit less severe before the transplantation. And also, the gastroenterologist of my center did manometry, gastrointestinal manometry, and again, I am not clinician, I don't understand what I am showing, but what they told me is that when you do this test, this patient, before the transplantation, had a kind of pattern, which is a completely pattern with signs or intestinal neuropathy and then no movement. But then after the transplantation, 2 years, I think, no, 3 years after the liver transplantation, there was some recovery, and which is an objective sign that the gastrointestinal dysmotility was improving in this patient. Okay, so this is what I can say about that. Actually, there was a consensus meeting some time ago, I think around 5 years ago in Bologna, in order to reach recommendations for the treatment of MNGIE. And then the recommendations were that, I mean, the treatment should be done as early as possible, just because of the reason I said before. If the patient is less degraded, because this disease is progressive, then you are stopping the disease in a better scenario. And then choosing between bone marrow transplantation and liver transplantation depend on many factors that are included in the patient, mainly the fact that you have a fully matched donor, and also if there is some liver involvement, because in many sometimes you have also liver involvement, so in this case probably would be more adequate liver transplantation. It seems to me that the whole experience tells us that the liver transplantation is a little bit less risky than the bone marrow transplantation, but well, this is something that is more detailed and more accurately explained in this report that we published 5 years ago. Okay, so now I am going to talk about what therapies are now at this point, and I think have a little bit more prospects in the future. And there are two therapies. I mean, both therapies have proof of concept in preclinical models that they could work, basically biochemical proof, because the animal model that we have don't recapitulate the disease, the clinical disease, but they don't have first-in-human testing yet. So we have to rely on the possibility that we can get a clinical trial or something like that. And these two therapies are the intracellular enzyme replacement that now is carried by Pierre-Pont Therapeutics, which is an enzyme, well, I will tell you now, I mean very little because I cannot say anything else. And then the gene therapy approach, which is something we have been working a long time ago. The first one is the intracellular enzyme replacement therapy. This is one therapy that was developed by a company called Entrada before and was taken by Pierre-Pont Therapeutics. This therapy seems to be working well in patients, in animals, and now they are looking for the possibility to do a clinical trial. So this is, well, a classical enzyme replacement in which you protect the enzyme, and then the enzyme go intracellularly, and then it would go, depending, I don't know the details, so I don't know to which cells it goes and to which cells it doesn't go, but the question is that you are able to provide the thymidine phosphorylase to the cells, and then intracellularly degrade the thymidine phosphorylase. So I hope they can get the possibility to do that, because I think it would be a good option to do that. And then the second one is the gene therapy. We have been working a long time ago on that. We started first with lentiviral vectors, and then in collaboration with Carlo Viscomi and Massimo Ceviani, we started also with AAV virus. This is the strategy with the AAV virus, which is providing with an AAV virus vector the thymidine phosphorylase with a liver promoter to the liver. So it's instead of doing liver transplantation, putting the thymidine phosphorylase in the liver of the patients. And then, as I said, in collaboration with Carlo Viscomi and Massimo Ceviani, we did this. We put the enzyme with a liver promoter, and then we tested in an animal model which develops the biochemical imbalances of the disease, which is increase of thymidine and deoxyuridine, but there is no clinical phenotype. And we tested several doses, and then we saw that these are normal mice, these are double knockout mice, and these are increasing doses in which we see that the effect is very good over the time, even over reducing. So we are putting a lot of thymidine phosphorylase in the liver of these mice. But as I said, these mice don't develop the clinical phenotype, so we try to stress the clinical phenotype by giving them oral thymidine and deoxyuridine over the whole life, and then testing the effect at the end of the life of the mice that have been fed with thymidine, with an excess of thymidine of deoxyuridation. In addition to the genetic defect, you are stressing giving more substrates. And then in this group of mice, we tested also the effect of the therapy. And then we could see here, in this case, it was more difficult to do a long follow-up experiment like in the previous one, because they are drinking nucleosides every day in a long time, so there is no, I mean, we could see better the effect doing pharmacokinetics, and in the pharmacokinetics we see that the mice treated, these are mice, exposed mice means mice that were taking the nucleosides in the water, and then we compare the levels of the exposed mice after treating with the vector, and then we see, and here we tested different promoters in order to see which is better, and we saw that the best promoter, which is AAT promoter, will reduce the peak of the pharmacokinetics, the area under the curve, and also the time to reach the normal concentrations. Okay, so this served us also to partially improve the vector. And then there were some, not clinical, but some less biochemical and more functional things. For example, accumulation of this white signal in the ventricles of the brain that was reduced. I mean, in the exposed mice, this was increased, and it was reduced with the best vector, with all the vectors in general, but with the best vector in particular. And also, not that clear, but because there is a lot of dispersion, motor function with the rotarod. I mean, the exposed mice had the tendency to be here, as you can see here, and the treated mice, well, I mean, it was significant only in some groups, but it seems that it worked. But I think the main point is a biochemical proof that it works. Then we have even improved more the gene because we remove the CPG sequences, which are sequences that are immunogenic, so removing the CPG from the gene is making the gene less immunogenic, and we tested this in comparison with the wild-type form in order to see if this modified gene was equally active as the one. It should be, because the protein encoded is the same, but in any case we tested, and then we saw that here, testing the wild-type form, which was demonstrated before in the previous experiments that work, and here reduce the timidine, but equally the modifying form also do that. And also the timidine phosphorylase levels that we reach in the liver of the modified form was the same as especially when you divide by copy number, is exactly the same as the wild-type. So the modified form is equally valid, and then we got. Well, before going to what we did, I want to use this chance to tell you something about the extent of the reduction of the nucleosides in the tissues, because we have seen blood so far. But I think it's important to notice that in all our experiments, we have seen that timidine is reduced in liver, as we can see here, in brain, in the skeletal muscle, but it's very hard to reduce in the small intestine. This is in the mice, in the previous experiments. And the same, exactly the same for the oxyuridine. I mean, the reduction in the small intestine seems to be harder to get. I don't know if this is related, and I suspect this is related with the fact that you are eating food that contains timidine, and then you cannot reduce it with the liver because it goes before the liver. In fact, we observe similar thing in the model in which we give a lot of timidine and deoxyuridine. I mean, here it's not so clear because there is more dispersion, but you can see here that here there is a tendency in the mice treated with different vectors reducing in all the tissues, but in a small intestine is basically the same. So we are not achieving, at least in the animal model, with this systemic reduction. I mean, there is one tissue, which is gastrointestinal, well, the intestine, the small intestine, which is very important in MNGIE, where we are reducing. So we have to put the light or keep the idea that probably we need to reach also the small intestine in order to be able to locally reduce this, because probably this is one of the reasons why intestine is a little bit more affected. I don't know, but I wanted to say that. And also this relates with the fact that or this could relate with the fact that when you do peritoneal dialysis, you see something, because probably locally you are doing something else than in the other cases. And then going back to the vector, and we had the chance to collaborate with a group of surgeons in the University of Cambridge, and then they did a transplantation of a MNGIE patient, and they took the liver of the MNGIE patient, and with a normothermic machine that is able to maintain the liver alive, they put one vector, no our vector, but one vector that was available there, because this had to be done at the time the patient was transplanted, and then they cut the liver, and then half of the liver was and was treated with the vector, and the other one was studied with the treatment. And then we can see here that the vector was able, in a human MNGIE liver, to provide timidine phosphorylase and to reduce the generation of timidine of this and the oxyuridine during this normothermic perfusion. So this indicates that this could be useful. I mean, this works in a human liver, in a MNGIE liver. Okay, so. And then we obtain orphan drug designation from the previous version of the vector and also for the current version of the vector. So we are close or we are near the possibility to do a clinical trial, but then we failed to get funding. But at the end, with the leadership of Gel van den Amil, we were able to apply for LifeArc for a grant in order to provide funds in order to do the regulatory preclinical studies. I mean, initially we wanted to do also a study in humans, a first study in humans, but at the end we get awarded in order to do the biodistribution and toxicity studies in rats, which is something that you had to do in GMP conditions in order to be able to demonstrate that your vector was well. So we got awarded, and then this will do, as I said, a study in rats in which we inject the rats with the first dose, which is the highest dose that you expect to give to the patients, and then 5 times more and 10 times more, and then see the biodistribution and the toxicity pattern of this administration in the rats. And then this has been awarded, so we have funds now in order to do the GMP-like production by a company of the vector, and then do the biodistribution studies. And if this demonstrates that in rats the treatment is safe, we will be at the point that we can do a clinical trial if we get funds for that. So this is already funded, and then we are happy because at least we will advance a little bit towards the possibility to do a clinical trial. So I think this is everything I wanted to say. Well, this is just the summary remarks. I think is the fact that MNGIE is a very well-known disease, and the therapy approaches should be aimed to lower the timidina and the oxyuridine, and then at this point we have the possibility to do bone marrow transplantation and liver transplantation, and what we have to do and what the clinicians have to do depends on the specificities of the patients. There are emerging potential treatments that we hope in the near future could complement what we have now. And then I think we have to keep an eye on the possibility that we probably need to be a little bit more focused on the intestine, on the local reduction of timidine in the small intestine. So this is everything I wanted to say. Probably I was a little too long, but well, this is the list, my research group, and this is the list of collaborators that have been doing something with this work. Okay, thank you.